Hi everyone, today I'm going to explain to all of you about vesicular transport. Vesicular transport is basically the transport of materials within the cell through the formation of vesicles. As you all know, vesicles are spherical membrane structures that are formed within the cell to fetch large materials. So when we talk about vesicular transport, they can be the taking in or bringing out of substances into and out of the cell. When we talk about the intake of materials into the cell, there can be endocytosis. There are two types of endocytosis that you need to learn. One type will be pinocytosis. Pinocytosis is the taking in of small fluid materials into the cell through the formation of vesicles like these. For the intake of larger materials, it will be phagocytosis. Another term for phagocytosis will be cell eating. Another term for pinocytosis will be cell drinking. So now I'm going to show you about the process of phagocytosis. The process of phagocytosis involves the cell coming near to a material that is large. Usually this material can be a foreign invader like a bacteria or food materials. So when the cell comes near this material, the cell will actually project cytoplasmic projections. These cytoplasmic projections are called pseudopods. Another meaning of pseudopods would be fake feet. So when these cytoplasmic projections are projected outward, it will encompass the whole material. Eventually, these two pseudopods would fuse with each other, and it will enclose the whole material, eventually forming a circular vesicle. This circular vesicle that's been formed will be called phagosome. So you can see this phago word through phagocytosis, the formation of the vesicle through the process of phagocytosis. Once the phagosome is being formed, it enters further into the cell, and in order to break down this food material or this bacteria, the cell will utilize lysosomes. Lysosomes contain the hydrolytic materials that will eventually fuse with the phagosome, and it will release the hydrolytic enzymes, thus allowing for the digestion of complex biomolecular materials within the bacteria or the food materials. After digestion takes place, the breakdown of the complex biomolecular materials will take place, and once simple substances form, these simple biomolecules can be assimilated by the cell. For undigested materials that cannot be digested by the cell, these will be indigestible materials enclosed in the form of a residual body. So in order for the removal of these indigestible materials out of the cell will need to undergo a process which is the removal of the substances from the vesicle out of the cell. So this process involves the fusion of this vesicle with the cell surface membrane, and this process we call it exocytosis. Once exocytosis takes place, the indigestible materials will be released out of the cell. And thus you can see the whole process of phagocytosis involves this whole range of structures, and you can see that endocytosis is the intake of materials and exocytosis is the removal of materials through formation of vesicles.